Hello everyone, my name is Hussain Ali and I am a PhD candidate from Department of Civil Engineering. Today I will be presenting my poster titled as Termite Hindgut Bacteria Tab 5 Key to Lignocellulose Waste Decomposition. We believe that bioenergy from lignocellulose biomass is one of the most promising candidates to help move away from the fossil fuel. The termites are commonly known for their ability to degrade wood, and the microorganisms present in the hindgut of termites are responsible to decompose lignin in the wood. Therefore, the overall goal of our research is to use Tab 5, which is the fastest growing microorganism isolated from the hindgut of Reticulotermis flavipes termite, to boost the methane production. Coming towards the introduction, we know that there are three major components of plant biomass, which are cellulose, hemicellulose, and lignin, as shown in the figure. More than 50% of municipal solid waste landfill in the US is comprised of lignocellulose, and the agriculture waste can have lignocellulose content up to 90%. Coming towards the problems that we are currently facing, the first and the major problem is lignin, which shields the other lignocellulose components like cellulose and hemicellulose and protects them from decomposition. This undegraded waste takes up valuable space in the landfill and also reduces the potential for production of renewable gas from the waste. The current methodologies that we have are either expensive or they create toxic byproducts. Our research has two main objectives. The first one is to determine the optimal conditions for efficiently growing Tab 5. For that, we need to determine the lag phase, log phase, stationary phase, and death phase for Tab 5. We need to see if it grows better aerobically or anaerobically. We need to find out the optimal ratio between Tab 5 and R2 broth. We also need to see what temperature it grows most efficiently at. And we also need to figure out what's the best way to produce large volumes of Tab 5 for field scale studies. Coming towards the second objective, which is to determine the optimal addition of Tab 5 to enhance the methane production. So methane generation rate depends on two factors: rate of waste decomposition and the rate of methane production. The rate of waste decomposition depends on Tab 5 and the rate of methane production depends on methanogens. Therefore, we need to figure out an optimal ratio for both of these bacteria to work efficiently and in synergy. Coming towards the methodology for objective 1, we first grew our Tab 5 cells on R2 agar plates and then we grow them at different temperatures. We found out that the maximum temperature that can grow these cells was at 37 degrees Celsius. After the successful growth on the petri dishes, we transferred our cells from petri dish to R2 broth, and then after the growth in the broth, we stored them at minus 80 degrees Celsius, and then we took our growth curves and we also did cell count to confirm that our cells were not dead. For the methodology of objective 2, we are using serum bottles as our batch reactors, and we are using rubber septa and aluminum seals to seal our batch reactors. We are using 4 different types of waste, which are wheat straw, rice straw, corn stover, and rice husk. We have used different ratios of microorganisms of Tab 5 and methanogens, which ranges from 0% to 90%. We have made in a total of 84 bottles, out of which 4 are our controls. We have added water in each bottle to achieve a moisture content of 90%. Coming towards the results, we can see here that the first figure shows the growth curve for the Tab 5, and from this figure we find out exactly at what time we need to use our Tab 5 cells. The second figure here shows the methane production based on different ratios. So we can see that without any use of Tab 5, we are getting lower methane production as shown right here as compared to using Tab 5 and getting a higher methane production. Our work is currently under the way, and right now we are testing for rice straw and wheat straw. Currently, we have concluded that we have a methodology to grow the TAF 5 at large scale, and we can also see from our preliminary results that there is a huge potential for increase in the production of methane by using TAF 5. And coming towards the acknowledgement, we would really like to thank US Department of Agriculture for providing this grant. Thank you.